good morning. We're going to start again, and we have the first symposium of this meeting, of which the topic is real-world evidence on clinical trials. Which should I believe? Now, as I mentioned in my previous talk, when I first went to the European Medicines Agency, clinical trials were the only thing that were considered important. And it was gradually the agency became aware that there was this thing that was known as real-world evidence. And there was also the debate going on as to whether we could actually believe anything that came out of pharmacoepidemiology, and to the extent that in 2010, the International Conference on Pharmacoepi in Brighton had a debate which was actually organised by the EMA, and the title was Results from Epidemiology and Pharmacoepidemiology Studies Mislead the Public because of a failure to adjust for multiple comparisons. Journals should not publish studies that do not account for multiplicity. And this was largely in a response to, let's say, multiple amounts of correspondence from Dr. Young to the EMA. And so we had Dr. Young and Professor Stuart Pocock speaking for the motion and speaking against this in defense of pharmacoepidemiology were Professor Sonia Hernandez-Diaz and Professor Ken Rotman. Now, we had actually stacked the stakes very much in favour of pharmacoepi by holding it at the pharmacoepi conference and having such eminent speakers. But Stuart Pocock somewhat cheated by being so entirely reasonable in his thing that we more or less had to agree with his premise that both were needed, and it wasn't down to a debate of one or the other. Now, I think we've come quite a long way since then. We still believe that both are needed, but there is to some extent still the, you might say, arguments that occur when the real-world evidence does not look exactly the same as what has been seen in clinical trials. So today we have a symposium, and I have 3 superb speakers who have agreed to take part and who are going to be giving us different aspects on how do we account for differences in the evidence, what we do when we don't have enough evidence and we're doing decision making, and then finally we're going to have a look at the Salford lung study, which was basically a marvellous real-world study on an investigational drug. But first of all, I'd like to invite Dr. Rassan to come and speak. Dr. Rassan is a pharmacoepidemiologist and technologist with over 25 years of academic and industry experience. He was assistant professor of medicine at Harvard Medical School, where he focused on methods for improving the quality and validity of studies based on real-world data, including admin claims and electronic health records. He is co-founder and president and chief science officer at Etienne, and I hope I've got the pronunciation right, I probably haven't, which is a New York-based healthcare technology company that provides science-driven real-world evidence solutions to healthcare companies, regulatory bodies and academic centres. So can I invite Dr. Rassan to speak on the use of real-world evidence to augment or replace clinical trials, a perspective on current research. And actually, can I ask all the speakers to get fairly close to this microphone, because as you've realised, we're having a lot of competition with the neighbouring session, and I think some of you were fortunate enough not to hear what I was saying earlier this morning, but I would hate to miss you hearing what my really good speakers are talking about, so thank you.
So, as Dr. Blackburn mentioned, I'm here to talk about the use of real-world evidence to augment or perhaps even replace randomized clinical trials. And this is a perspective on current research. So I'll spend quite a bit of time looking at some of the studies that are comparing and contrasting these two methodologies and the results that they've gotten to date. So as was mentioned, I'm an employee of and a co-owner in a company called Adion Incorporated based in New York City, and this talk was developed in collaboration with my colleague Sebastian Schneeweiss. So before we talk about real-world data versus randomized clinical trials, I wanted to at least put one or two slides on what is real-world data, and then a corollary, what is real-world evidence, RWD and RWE. And using this particular definition from the U.S. Food and Drug Administration, real-world data are data related to patient health status and or the delivery of health care routinely collected from a variety of sources. And in the introduction, some of those sources were mentioned: administrative claims data, electronic health records, but you could also think about wearable data, perhaps even genomic data, other kinds of data as well. And real-world evidence, RWE, is the clinical evidence regarding the usage and potential benefits or risks of a medical product derived from the analysis of RWD. So for me, that's a useful working definition of RWD data and RWE evidence. So I wanted to show a couple instances of use cases of real-world evidence in regulatory decision-making. Case 0 here, numbering from 0, is the use of real-world evidence in the primary approval of a drug. And some examples are the use of historical controls or synthetic comparison groups. And I was thinking about this particular line on this slide, Stella, as you were introducing and talking about that symposium from 2010 and how there isn't necessarily a total difference between randomized and real-world data anymore. It's sort of come together in some ways a little bit as well. A more common use case, I would say, is secondary indications, using real-world evidence to support the secondary indication for a drug, such as an indication expansion, population expansion, say expanding to a pediatric population, or an efficacy claim expansion. In the EU, very frequently, RWE associated with adaptive pathways, using RWE directly after an initial approval to refine and better understand the performance of a product. And then I'd say some more classical use cases for real-world data, real-world evidence, safety either as a post-market requirement or rapid response to a safety signal. These are all places where we've seen real-world data and real-world evidence used in these contexts. But how do we take real-world data and make it real-world evidence? There's a gap, certainly, between the two. And so for inspiration, I want to spend a few minutes going through about 5 studies where we have examples of real-world database studies that have come to the same conclusion as an equivalent or near equivalent randomized trial. So, first example here is a real-world data cardiovascular safety study preceding an RCT. And this analysis here was blinded to the RCT results. And you can see the study published in Arthritis and Rheumatology by Soyoung Kim and colleagues. The hazard ratio for composite CV event was 0.9 with a wide confidence limit, certainly including the null. A randomized trial looking at the same question was published, if I recall correctly, several months later. That was the OnTract trial. And they found a hazard ratio for composite cardiovascular event of 1.05 with, again, a confidence limit that included the null. So my interpretation of this is that these are very substantially similar results, and from a regulatory perspective, results that would lead to the same regulatory decision. And ultimately, for me, that's the most important criterion. Another example, risk of cardiovascular events in older patients with gout, initiating febuxostat versus allopurinol, population-based cohort study by Zhang et al. Risk of composite cardiovascular outcome 1.02, looking in comparison to a trial published in the New England Journal of the same topic, the CARES trial 1.05, again, both including the null and thus leading to the same regulatory decision. The EMPA-REG study, in this case going the other direction, a randomized trial on a particular population looking at empagliflozin, cardiovascular outcomes and mortality in type 2 diabetes. They saw a hazard ratio of 2.9. This was not the primary endpoint of the study, but a hazard ratio of 2.9 for that drug, empagliflozin, and the risk of diabetic ketoacidosis. A follow-up real-world data study was conducted, also published in the New England Journal by Freilich et al., looking very specifically at that issue that had been noticeable in the published trial results, and found a hazard ratio of 2.2, so indicating to the same potential concern about DKA in this agent, but very, very low absolute risk, which is a good thing, clearly. And then one or two more. The RELY study, dabigatran versus warfarin in patients with atrial fibrillation, stroke prevention hazard ratio of 0.66, not including the null. Real-world data study done by Seeger et al. Hazard ratio of 0.77, but with a wider confidence limit. So a perhaps directionally similar, but maybe slightly differing result. And I have other examples here as well. This one is about a secondary indication for an oncology drug versus chemotherapy for ALL, indicating in both cases efficacy. So when we look at all of these data, perhaps there's a feeling, or I don't want to use the word bias, but a tendency in the audience, perhaps following on from discussions like that 2010 discussion in Brighton, in that very, very, very dark hotel room, or hotel ballroom, as I recall correctly, about how much a priori trust we would have in an RCT result versus a real-world data result. And I would argue that this potential difference in a priori trust isn't the same for all studies. We can think about unintended effects and intended effects. We can think about beneficial results and harmful effects. And just a schematic here, I won't walk through it, indicating perhaps an average difference in a priori confidence. And I've labeled here some of the studies I showed you as to where they would appear on this diagram. So the question is, can we achieve equivalently sized bars for real-world data and RCTs in the two categories on the top row, beneficial effects, and can we even exceed RCTs looking at unintended harmful effects, meaning by and large safety studies? So the big questions, you can tell they're big questions because it's big letters on the screen here. Why are some real-world data studies able to come to the same causal conclusion as RCTs? And a parallel question is, are some not? And if they're not, why not? How can we ensure that the next real-world data study that we do will continue to come to the same conclusion as an equivalent RCT? And then how can we know if we, as a regulator or as otherwise an evaluator of that information, can make a decision based on those conclusions? And I want to be clear. There are a lot of RCTs in this world, and the vast majority of them are best done as RCTs. So if you have a pivotal study for a new drug that is coming into a market with a clear standard of care, that sounds like an RCT to me. There isn't going to be real-world data, very potentially, that's going to answer that question in a satisfactory manner, especially if it's a new agent and there is no real-world data on that agent. But to the right side of this spectrum, where RWD analyses become possible, and all of those studies I showed you are cases where our RWD analyses become possible, this is what I'm talking about, and it's 20%, 25%, maybe 15%. Let's talk about where we could supplement or even take the place of RCTs in these cases, and these cases only. So, as was mentioned at the earlier session today, my colleague Sebastian Schneeweiss proposed a framework, MVET, which I've switched a little bit here, but it's the same idea. So Sebastian's framework was M, meaningful, let's ask meaningful questions. Valid, and that's high on both of our lists. Let's make sure that the way that we're doing the study is of very high quality and very high validity. E, expedited, and for me, reproducible and expedited are very much linked together. Let's do it quickly, let's do it in a way that others can, in an expeditious, quick manner, reproduce. And let's be T, transparent as well. So let's, if any of you saw the discussion I did yesterday about visualization, that's one method, but many other methods are out there as well for being totally T, transparent at all times. And I won't go into more detail this morning, but happy to talk at a break about this framework. What I wanted to do instead was to go into more detail on a single case study of a case where this is Schneeweiss et al. Considered using real-world data for a study that had been using real-world data for a randomized controlled trial that had been used to support the application for a secondary indication for telmisartan, Mycardis, in 2009. So this was a drug approved originally in 1998 as an antihypertensive. There was a supplementary indication given in the US in 2009 for cardiovascular risk reduction in patients over 55 years. And that supplementary indication was supported by the OnTarget trial, which took approximately, if I recall, about 6 or 7 years at a cost of roughly $300 million US dollars. And the OnTarget trial compared telmisartan to ramipril. Ramipril had a known cardiovascular benefit, and so the idea here was to demonstrate equivalent cardiovascular benefit to ramipril, looking at cardiovascular death, myocardial infarction, MI, stroke, and hospitalization for heart failure. And so the thought exercise here, and the study here, was to rewind time and to pretend that we were starting this trial in 2004 or 5-ish, but instead of doing it as a randomized trial, to use real-world data, and to see if real-world data would have led to the same conclusion that regulators could add a second indication for cardiovascular risk benefit in patients less than 55 years old. And so how do we do this? This is the MVET framework, it's a meaningful question. Let's use valid methods. I'll talk about that in a moment. Let's do this quickly. And this was about 16 weeks as compared to 7 years. And let's do this transparently. And the publication that supports this, again by Schneeweiss et al., very transparent in the methodologies used. So you can see here that this is telmisartan versus Ramipril. Unmatched, you have your unmatched data on your left here. Some substantial differences in these two populations, indicating need for strong confounding control. That confounding control was achieved through propensity score matching, so a valid method in this case for achieving confounding control. And after matching about 4,665 patients in the Ramipril group, the same number in the telmisartan group, mean age of 69 years and change in each of the two groups. And good balance on all of the meaningful confounding factors for this study. I won't go through this for time, but you can inspect the slides later on. And looking at the composite endpoint, which as I mentioned included stroke, MI, hospitalization for heart failure, the observational study, this real-world data study, found a hazard ratio of 0.99, indicating equivalence or similarity between these two agents for their cardiovascular risk-benefit. And 1.01, a very substantially similar result in the on-target trial. And the real-world data was done, as I mentioned, over the course of 4 months instead of 5, 6, 7 years, at a cost clearly substantially less than $300+ million US, and with a result that's very similar. And let's look at some of the specific endpoints. Stroke, relative risk, 0.95 in the RWE study, 0.91 in the clinical trial. MI in the middle here, 0.92 and 1.07, so a little bit different, but again, both including the null. And heart failure, 0.95 and 1.12, again with those confidence limits that include the null. Now, what you can see here is that this answer was intended to be null. But we do know that there is a difference in these drugs, that telmisartan should be protective against angioedema. It's a known side effect of the comparator drug. And so in the same study, they also checked to make sure that that negative control, or that known value was also represented. And you can see here 0.13, strongly protective effect against angioedema in the observational study, and 0.4, again, a very strongly protective effect in the RCT. So the way this was carried out certainly represented those themes of MVET, meaningful, valid, expedited, and transparent. And so the question I posed at the beginning is, how do we know when this could work? And we have a series of questions to ask ourselves, and this is where I'll end. Is the setting adequate for RWD analysis? That was that bar where we selected a certain set of cases that were adequate. Is the data quality fit for purpose? We have to make sure that we have the data available to answer the question. Do we have a statistical analysis plan that's clear and transparent? Do we have a plan to address any potential bias that we could find in our real-world data? And do we have a high-quality analysis with transparent and structured reporting. And as we continue to develop evidence, do we have a way to make sure that we have an answer now, but also an answer that updates over time as our understanding of the clinical picture, or indeed the use of the drugs in the population, and thus the performance changes over time. And with that, you can get valid, transparent, reproducible results and do it quickly. So, thank you.
It's now my pleasure to invite Professor Deborah Gert of the School of Pharmacy and Medical Sciences, who is also Director of the Drug and Therapeutics Information Service and past President of the Australian Pharmacy Council, to give a talk. And Deborah has worked extensively in academic detailing and on the quality use of medicines and medicines policy. She's a member of the National Drug Utilisation Subcommittee of the Australian Pharmaceutical Benefits Advisory Committee. And Deborah is going to talk to us today on the practical problems of deciding on recommendations for drug use when there is mainly clinical trial data and we know that the real-world evidence will be different. Thank you very much, Professor Hurd.
Thank you very much. And in this presentation, what I'm going to focus on is to follow on from what Dr. Blackburn was presenting in her plenary this morning, and that is that there are a number of stakeholders involved and interested in this information, and it is a process across time. It's not just an initiation or introduction into the market of a drug that we are grappling with this decision. So I wanted to share with you the experience that we've had in Australia and give a couple of examples. In Australia, and my presentation is coming both from the payer and also from the clinician. So in Australia, the payer is the Australian Government subsidy scheme. There are other payers, but the Pharmaceutical Benefits Scheme, which is established under our National Health Act. And under that National Health Act, it has the responsibility of establishing our statutory expert advisory committee, which is our Pharmaceutical Benefits Advisory Committee. And the primary role of that committee is making recommendations to our Minister for Health regarding those medicines that will be available for public subsidy. Since 1989, however, we've had available community prescription data that is available to the Drug Utilisation Subcommittee through our National Department of Health. And DUSC, as I'll refer to it going forward, they collect, analyse this information that is acquired through the claims made for those medicines subsidised through our national scheme. And its role is to make recommendations to our payer, the PBAC, or the advisory group, to our government, both around comparative effectiveness and also cost. And the government has developed guidelines for preparing submissions to our Pharmaceutical Benefits Scheme. And I think this was picked up a little bit in Dr. Blackburn's presentation, is giving this advice to people around the information that needs to be presented for making a decision. And this was updated and published last year, September 2017. There's been many versions. But what I wanted to highlight, that in this submission structure for a major submission for consideration of a medicine for public subsidy, it includes this Section 4. And Section 4 is the use of the medicine in practice. And it presents the most likely extent of use and financial estimates. But this is quite challenging because where we've got the efficacy studies, we've got the clinical trial information, we've got what might be predicted is going to be the uptake in use. But again, Dr. Blackburn highlighted this morning an amazing example of what was expected, and then within the first week, 100,000 males were using that new drug. And we've seen many examples of that as well. When new medicines may become available, it there is some uncertainty as to how are the clinicians going to use that medicine in practice. And so this is a very important part, this section 4, is understanding that use of medicine in practice. So the sponsor is asked to present what they think might be the most likely extent of use based on their clinical trials, the work they have done in their development of that drug. But estimating the likely uptake of the proposed medicine and the cost to government is really a critical part of this section 4, and the Drug Utilisation Subcommittee provide advice then on that section 4 estimates. Both epidemiological and market share analyses are the two broad approaches used for developing utilisation and financial estimates, although their use is not mutually exclusive. I just wanted to highlight in this infographic, where we're talking about epidemiological and market share. The epidemiological approach, as you might expect, uses incidence or prevalence data to estimate the number of patients treated and units dispensed. But there can be challenges because having the epidemiology available for some of the new conditions, you mentioned the breast cancer example, now there's subsets of subsets, and the epidemiology is sometimes not there for estimating. But also we use the market share approach, which uses current market data to estimate the market share, the number of patients treated and the number of units dispensed and market growth. So these form the two principal approaches. Clearly there are other people in the world grappling with this challenge as well of trying to forecast what drug utilisation will be when it gets into the real world. What's the real world experience? How is it used? So this is a formalised process, and Drug Utilisation Subcommittee reviews all of the medicines on our schedules and that have been listed for 2 years, and select individual medicine groups to look at them and review them. Also there might be ad hoc reviews, and I'll come on to give some examples. But there might be planned, the planned reviews are listed on the PBS website. One of the features of this presentation is around transparency. And sponsors are notified at least 10 weeks before a DUS meeting of the report, and they can give some feedback. Sponsors are provided with the DUS reports on the utilisation of medicines 4 weeks before a DUS committee meeting, and each sponsor has 2 weeks to provide a written response to the DUS Secretariat. Just moving on. One of the things that has changed that I wanted to highlight is that the publication of the DUS reports, which started in 2014, is now a routine process. So public release summary documents are now published on the Pharmaceutical Benefits website. And I just wanted to give a couple of examples and walk you through those. Dr. Rassin has highlighted the real-world evidence and also the clinical trial evidence around the anticoagulants. And so the new anticoagulants, we looked at this, and this is one from our public summary document that is available on the website. And what I wanted to highlight here were the key findings, which was based on the volume of prescriptions dispensed. So the NOACs contributed to a growth in the anticoagulant market. And in the calendar year of 2015, for NOAC versus the warfarin use, there were 1.6 million prescriptions supplied to 188,000 patients. And for 72,484 patients, this was their first NOAC prescription. So the purpose of this analysis is to better understand what we might have predicted was going to be the utilisation. So how was it going to be used in the market, to how was it actually used, and what were some of the reasons for the differences between what was predicted and actually actual use. What was the real world experience and use? DUS considers a range of factors in its analysis, and it considered that co-administration with aspirin would not be one of those analyses because it's so routinely available over the counter, it wouldn't be captured in this data. So if we look at the data, we can see the blue, the very large blue here, that represented the warfarin, and you can see that's declining, and you can see the index point where NOACs were listed, and also you can see the patterns of use where rivaroxaban, dabigatran and apixaban. I know we already pronounced them slightly differently, but if you forgive that. And what we saw there was that also by age group, and looking at the rivaroxaban, and this reflects the utilisation for principally different conditions, and whether it be for atrial fibrillation or not, you can see here in the older age groups. So we've got rivaroxaban peaking in the 60 to 75, and this was reflecting its indication on the PBS for other conditions other than atrial fibrillation. But what I wanted to highlight was here now by the states and territories of Australia. So if I highlight two states in particular, Queensland and South Australia, where I'm from, so we have the same we have the same clinical trial information, but in those two states where their hospital systems have clinical formulary, where they have hospital formularies, where one or two drugs might get on the formulary for managing atrial fibrillation. So the people making those decisions in Queensland, they decided on rivaroxaban for their hospital formulary, and in South Australia they decided on apixaban. So you can see the patterns of use in our two states reflect different decisions made on the same evidence, and that then changes the patterns of use in those two states. There are probably other examples, but it was just to highlight that the real world experience and use by clinicians can be influenced by other things in that environment. And here's another example, which was around ferric cup oxymaltose, which was a 24 predictive versus actual analysis. And again, just wanting to share with you some of the key findings. This was recently listed in 2014, and you can see here when that was listed on for public subsidy, and increased utilisation in the country of Australia. But what we can see when we look down at that is that the biggest increase in use of ferric cup oxymaltose was around the use by our general practitioners in primary care. But what are some of the reasons? And this is the challenge always to try and understand what's influencing, sort of addressing some of that uncertainty for decision makers. And when we looked at the different utilisation, the community use represents the greatest increase in the volume, but the highest rate of growth has been in our public hospitals participating in our pharmaceutical benefit scheme reforms. And this might reflect patient blood management strategies of the hospitals. So it might not just be about how the drug is being used, it might be changes in how in this case, how blood is being managed. So better management of iron deficiency anemia may lead to less requirements for use of blood, for example. So the objective of predictive versus actual review is to provide general insights to improve the process of preparing future estimates of drug expenditure for sponsors requesting listing of medicines on our pharmaceutical benefit scheme, and also to identify quality use of medicines or other problems that may be referred to the PBAC for further consideration. So this process is, and there's many now that have been done, and and they're published on the PBS website. But they're starting to inform and improve our understanding of where some of those uncertainties are, to improve our methods of coming to understand what might be predicted utilisation based on the evidence that's available at the time of listing, after it's gone through the regulatory process, to how it is used in practice by clinicians. And that use by clinicians in practice will vary because of the context in which they're working. It may be because of statewide formularies or other access requirements, but it might be because the clinical guidelines in the country change, and therefore the utilisation might change in parallel. And so this is a process that we have been using now for a number of years in Australia, and I think it's we're coming to understand again where those areas of uncertainty are and how we can use that to better forecast the utilisation and to inform decision making, to reduce some of the uncertainty in that decision making. And I will conclude there. I think I'm just on time. So thank you very much.
And now I'd like to introduce our third speaker, Dr. Andrew Rodham, who is currently Vice President and Head of Epidemiology, Real-World Evidence and Digital Clinical Platforms at GSK, and is an expert in epidemiological research. And Dr. Rodham is going to talk on the Salford lung study, how a study with an investigational drug was carried out in a usual care setting. So giving a third example of how we can use this. Thank you, Dr. Rodham.
Thank you. So what I'm going to do is tell you the story behind what we did at GSK, actually before I was even there, back in 2010, 2011, when we started out to do this study, to look at how you can design and implement a clinical study of a drug which was an investigational medical product at the time, but to do so in a much more usual care setting. And I sort of want to start a little bit by why was that something that we really wanted to do? And I think it goes back to where Stella started this morning of the traditional world of it was always the clinical trial or it was the epi study. This is somewhere in the middle between the two areas. And if you think of what it is we actually want to know about medicines in today's world, ultimately, when you think about getting a drug efficacy, what you're looking at is measuring the effect of a drug in a really ideal clinical setting. It's the setting which has the most cleanest, nicest patients you can imagine. They don't have comorbid conditions, they don't have co-medications. They do everything you want them to do. You can measure everything you want to measure on them. You encourage adherence by almost getting them into the clinic every time you need them to take a medicine. And you ask them how many times they're taking it, and you make sure that you tell them how many times they're taking it. And that helps you answer that very, very fundamental regulatory question of does the medicine actually produce the intended effect it's hypothesized to produce? The reality is that when you're a clinician or when you're a patient or when you're a payer, what you actually want to know is, does the drug really work in the typical setting of care that you will see? And the typical setting of care is not the patient who gets the regular adherence, visits, checkups, phone calls. It's not the patient who has no comorbid conditions, who doesn't have co-medications. They don't come in and get all of the things measured on them that you want to measure on them, so you actually don't necessarily know how effective the drug treatments are. And what we were thinking of when we were starting to design this particular medicine combination, I'll come on to why this was then the right study to do, is to think about, well, how can we truly measure effectiveness here? And in the world of respiratory medicine, this becomes even more important. So if you look at multiple publications in the literature of looking at how representative are clinical trial patients in phase 3 clinical trials across all sponsors, GSK is as bad as everybody else here. Roughly in asthma you get about 3% of patients, typical patients, appearing in clinical trials. In COPD it's about 7%. So that less than 1/10 of your treatable population appears in your phase 3 clinical trials. And there's some very good reasons for that. In asthma, the vast majority of patients in the areas we do clinical trials are in the younger age groups. Younger age people are very difficult to recruit into clinical trials. It's very difficult to keep adherence in there. COPD patients, they're old, they have many other comorbid conditions. They're never the sorts of patients that you can get that have this nice profile for our trial. And ultimately, if you go, again, think about how you do clinical trials, you go to high-end clinical centres who have got centres of excellence in that particular medical discipline you're going for. So you recruit patients from respiratory centres and respiratory medicine clinics. Not that many patients who have COPD and asthma go to a respiratory medicine clinic very often. Many of them are treated in the community, and therefore you don't see them typically appearing in your clinical trials. So when we were thinking about this new medicine that we were developing back in sort of first part of 2010, 2011, and we started to think about what evidence were we going to need to be able to demonstrate on it, this started to become a real issue that we wanted to think about. How do we get this more representative, usual care setting, how patients will actually use that medicine in practice? And 2 or 3 little things about the medicine which are really important to decide on the design of this study. So what we were developing, and actually the singular reason why this medicine was possible, or this study was possible, was we were combining 2 already licensed medications into 1 inhaler. And we wanted to know what was the benefit of giving a patient 1 inhaler versus giving a patient 2 inhalers. And then what was the benefit of giving a patient 1 inhaler that they use once a day versus the benefits of giving patients 2 or 3 inhalers which they use 2 or 3 times a day. That is, I challenge any one of you to be able to come back to me and say, how do you bring a blinded clinical trial which will allow you to answer that question effectively? It is an impossible design to actually do, and if you do a clinical trial, you will ultimately get the wrong answer of the question that is most important and valuable from this particular case. You can ask an almost parallel question if you're developing an injectable versus an oral therapy. You just cannot do trials which blind the patients or blind the physicians or blind carers to what it is going on. So we really wanted to focus on, well, if you get people to take a drug less often and only once, how effective is that medicine going to be? There began the birth of the sulfate lung studies. We did 2. Most people should have only probably done 1, but we did do 2. We did 1 in chronic obstructive pulmonary disease, and we did 1 in asthma. This is just the COPD description. But we wanted to know was how effective was this new medicine going to be compared to usual care? And then ultimately, how could we then design that study in the simplest possible way which replicated usual care as much as feasibly possible? So we did the simplest things that we could do. We said, basically, the doctor said the patient had COPD, they were over 40 for particular reasons of age definition of COPD in the 40s is technically potentially a different disease. They were taking medicines which suggested they had COPD already. They'd had an exacerbation in the previous 3 years, and they agreed to take part in the study. That's a relatively minimal set of inclusion criteria. We didn't require any actual formal verification of the diagnosis. It was purely down to the doctor to say they were. We then randomized them, they got the medication that we were developing, or they got their usual care, no specification on usual care, whatever they were being treated with. And that was it. That was the only time we saw them for a year. We then saw them at the end of the study and did an end of study visit. Everything else was how a doctor would usually treat that patient. The infrastructure you need to put in place to do that is quite substantial. So, remembering this was an investigational product, it was an agreement with the UK regulator, with the MHRA, with NICE, because this was being developed with effectiveness and submission for cost effectiveness in mind. I think I should probably also add, we weren't doing this study as a primary regulatory study. We were doing our traditional approaches of clinical trials for the regulatory approach. This was being supplemental to that, and was actually being the only regulatory impact of the study was to discharge a safety risk that we had that was a concern of putting 2 medicines together in a single inhaler. So why did we do it in Salford? And probably most of you in the audience are wondering, where on earth is Salford? I wondered where Salford was when I first joined GSK and someone told me I was going there. For those of you who don't know, think Manchester United, and where Manchester United play football, that is Salford. That small area around Manchester United's football ground is the borough of Salford in Greater Manchester. And the single reason we did the study in Salford was it was the only place in the world that we found at that point in time, 2010-2011, that had a fully integrated, connected electronic health record system that linked both primary care, secondary care, and some level of tertiary care in one single system that we could connect together. And how did we go about doing it? It was never going to be possible for us to do it as a pharmaceutical company on our own. It was a major collaboration. It was a collaboration between lots of groups of people that had to come together. There was all of the primary care doctors who essentially were the recruiting physicians. We had a network of up to 600 at any point in time who were recruiting patients into the study. We built a clinical supply chain that delivered medicine into pharmacies so that patients could get a prescription for their investigational drug and go to any pharmacy dispensing in that area to get that dispensed medicine. We built a connection to all of the tertiary hospitals in that area because we needed to know when patients ended up in a hospital. We had a huge collaboration with the National Institute for Health Research in the UK, who are the academic NHS bodies who help run clinical trials. There was a massive informatics component, which I'll show you on the next slide, and a whole range of academic, and clinical pharmacy experts who all came together to work as a single team to deliver this actual study. We put in a lot of governance, a lot of outreach to various different people, and it was a never-ending task to make sure the study was successful. In terms of how did we do it with the data flow itself, this is the really simplified version of how we did the data. We had on every single patient who was in the study, we had 14 different flows of data from different databases in the UK. Because remembering this was a study on an investigational drug, we had obligations to the regulator for expedited reporting and regularised reporting of every adverse event. The study was being conducted under good clinical practice. And if you also remember, we weren't assessing anything that happened to a patient. We could only look at what happened to them electronically. So we had their primary care records, we had their main hospital records, we had the out-of-hours service, so if they went somewhere overnight. We had nurses and primary physicians. We had national data flows if they went to a hospital that was out of area. And all of that came together so that every day, every morning, the study safety team would look at the data coming in, make assessments as to whether or not an event was an adverse event, and if it was, ultimately expeditiously report that to a regulator. Finally, all of that data came together 2 years ago, I guess it was now, and 1 year ago for one of the other studies, the AFMA study, into an analysis data set that was ultimately reported out. And it was joined together with a few other data sets that we collected independently. To go into what it was then in terms of the study itself, we ended up recruiting 2,800 COPD patients, 4,200 asthma patients. That's a significant amount of people in one tiny community area in a place in England most of you have probably never ever thought of visiting. We had 130 pharmacies dispensing medicine. We had 80 GP practices recruiting, and there's an average of 3 to 10 GPs per practice, depending on the practice size. There was, as I said, the one electronic medical record system that we followed. We ended up training more than 3,000 individuals in Greater Manchester in good clinical practice, because anybody who had to touch the data had to be trained. And as of the last estimates we ever have, we have somewhere in excess of 235 million rows of data. Every single encounter that happened to that patient electronically while they were in this study was something that we collected and followed up on. And it allowed us to really have the insight of being able to say, well, how can you do this study? How is it that we can really understand patients in their usual care setting by minimally touching them and following them through their electronic record? Both studies have now published. I think we published the COPD one first. It appeared in the New England Journal. The AFMA study slightly later in the Lancet, they recruited slightly different rates. Both of them showed the medicine being effective, and both of them showed comparable, but if not a bit like what Jeremy was talking about, the results aren't identical to the clinical trials, but they are directionally equivalent, and you start to understand where the differences might occur from. So we had two very, very significant studies which have helped us truly understand how you can do these types of study in routine care. And ultimately, this was the very first type of study in the world. It kept the rigour of the science behind it. We did have randomisation, and randomisation really helped us in understanding, at least at that first point in time, you can compare those two groups. We had an active control, there was a robust primary endpoint, it was a clinical exacerbation by definition that we could follow. The underpinning effort to make this study happen is absolutely enormous. That we employed as a company more than 300 people to make this study happen. We actually opened an office in Manchester to make this study happen. The significant effort to go behind making this happen was very, very tremendous. But it helped us learn about what could you do differently next time. How can you do these studies more routinely? What is it that you really need to do? Because I think what we believe, and I think what's been shown by the study, is that you can get some really critical information for healthcare professionals, for decision makers, for patients. And ultimately that will help us design better studies in the future, which help to bridge that gap that I think Stella opened this meeting with, on what is it that we really want to understand about medicines to truly design medicines which are effective for everyone in the future. And with that, thank you very much.
Thank you for a wonderful session. And I am wanting to ask two questions and ask for some panel discussion. The first is the question is for Dr. Grasson. There was one of those studies which you showed the equivalence and between real-world data and a randomized trial, in which the crossing of the null was not the same in both the randomized trial and the real-world data. We have all been trained as clinicians to think about the less than 0.05 as a really critical issue. Clearly, this is not so much an issue to you, and it would be very helpful for me and perhaps others to hear you explain the reason you included that particular example amongst the 4 or 5 very convincing examples which supported the proposal that real-world data can in fact potentially, in the right circumstances, replace a randomized trial. It is this question of p less than 0.05 or the crossing of the null which actually would be helpful, I think, for all of us, including me, to hear you discuss the reason you included that particular study in the examples which you were showing quite convincingly otherwise that there was actually equivalence between real-world data and randomized trial. Thank you.
Stand up so I can see you. Thanks for the question. I'm going to be a little philosophical in the answer, and return to the notion that Andrew mentioned of directional concordance or directional indication versus perfect or exact concordance. And I don't have the numbers exactly in my head. I could go look at the slide. But to me, they were indicative of concordance. Now, if the p-value is greater than 0.05, or the point estimate, for example, is below the null, but the confidence limit includes the null, that to me is an indication that more data could be helpful in order to really refine the answer. And to me, that's a very important distinction between our abilities to do research with real-world data versus a randomized trial, where we can actually then seek more data, either through the accumulation of time and more experience, or through the utilization of alternative data sources to help understand the question or the answer to the question more specifically or more concretely. And so, to me, that's a very fundamental distinction, is that real-world data gives us the opportunity, with appropriate statistical techniques about multiple looks and so on and so forth, to refine our understanding of an answer over time. And I think this speaks to your presentation as well, of how the dynamics of introducing a new drug in the population have both a clinical impact, but also a population impact as that population gets newly treated.
No, I think the only point I suppose I would go back to is fundamentally p-values come from experimental designs. And there's been a long debate in epidemiology about whether p-values are even valid in epidemiology because there's not an experimental design. And I think Jeremy kind of alluded to it. You just double the amount of data and you change the p-value. Well, is that really meaningful? The world where we can get more and more and continually adding and accumulating evidence, it's probably more the size of the effect and where it is that's the important thing to focus on, and less on maybe what the size of the p-value is and where it is. And I think when P's greater than 0.05, you're essentially looking at random number draws around a confidence. So the fact that they dot about above and below is probably in line with what you would expect them to do, certainly from my long history of statistical training a long time ago.
Thank you very much for that. And I'm wondering whether Stella yourself would like to also explain the reason for your expressed disdain for randomized controlled trials in your first presentation.
I think you need randomized controlled trials quite often just to give the indications, is there any efficacy in the drug under the ideal circumstances? But I think after that it becomes less useful because of the reasons we discussed earlier of the change in population from when you're going from the clinical trial situation to real world. And as I say, I think that we are always trying to reduce, to some extent, the confidence intervals around our estimates at any stage. And I would agree entirely that the p-value is not desperately important. It's probably the confidence interval around it that matters. Thank you. We have another question. Good.
Hi. I'd like to thank the presenters for great presentations.
So can you speak up more? Is the mic working? Sorry. Thank you.
I'd like to thank the presenters for excellent presentations. And my question is for Andrew Roden. Now where there's a lot of interest in real-world evidence in regulatory decision-making. So can you tell us how the Salford lung study was perceived by regulators, and did it end up in the label? That's my first question. And the second part of my question is basically if you can tell us the learnings from the Salford lung study in terms of doing real-world evidence. I know you mentioned it was a great effort, so should other pharma companies be thinking in the same direction, or this is something that we shouldn't be thinking about? It's just logistically a nightmare and it's not very feasible, so to speak. Thank you.
Thanks. So I sort of alluded to maybe what the regulatory position was during the talk. So when in our very early discussions with the regulators, the feedback was this, and our positioning was this was never going to be a study that was being used for regulatory submission. It was part of a discharge of a particular risk. There was a concern about pneumonia and it was used as part of that discharge. Not surprising, when we went back to the regulators with the results, it didn't change the label because of the a priori positioning of how the study was actually going to be used. But I do think one of the things that the study has probably helped drive more with the FDA than anyone else is how these types of pragmatic, usual care designs can be thought of for things like label expansion studies, where you may already have an indication and you may be wanting to expand the label. So I know I've given quite a few presentations at FDA board meetings. I know other colleagues on the study have been asked by the FDA quite a few times about what did we do, what did we learn. Which I think then builds to the second point of, how do you make these more routine? This was an expensive first proof of principle that you could do it. I think what we've done and what we're doing now is we've realised you don't have to tick every box all of the time. So we're now looking at how we do this in multiple sites, and we're just being very pragmatic about that and saying, well, in some cases we may not have an electronic medical record, so we'll use the traditional case report form. In other areas we might have registries, so we'll use the registry. In other areas we'll have an electronic record and we'll use that. But I think it's helped us a lot think through, well, how can we make it easier for patients or physicians to take part in studies? And some of it is coming together around even ICH, who are looking at guidances around how do you make GCP more usable in these situations, because one of the biggest challenges we had were how do we follow GCP guidelines and implement the study in a really simple and practical way. So I think we've learned a lot about just a lot of the operational stuff, and then dialing back some of the pragmatism we put in to say, actually, we don't need to be as pragmatic as that and still get a really good answer to the question.
Can I bring a thought a little in the same vein, but there's a project going on in the United States, commissioned by FDA, and for disclosure, I'm working on this project, so I have some conflict there. But it's part of the 21st Century Cures Act, where the FDA is looking more broadly to think about how RWE can be part of decision-making, that they do regulatory decision-making. And so what they've asked the investigators to do is to reproduce 30 clinical trials in the same way that I showed you with the telmisartan example, but at larger scale, to really understand in which cases, and Frank, this speaks to your question as well, in which cases real-world data can serve in the place of clinical trials, and equally importantly, in which cases it can't, and what differentiates those two cases. So this is undergoing right now. It's, as I said, 30 trials being reproduced in real-world data, as though they were run in real-world data at the same time as the original trial. And it's a nice mix. It's 23 trials that resulted in a positive decision by FDA, and 7 trials that resulted in a negative decision, to really understand this topic much more broadly, and taking into account all the different kinds of approaches that are available to us.
Thank you. Yes, please.
I'll just have a follow-up question to, whether observational study can lead to a similar result to a clinical trial. It's a little bit frustrating for me to believe that, only based on one study that you have similar result from an observational study with the clinical trial, then, so to me that could be just purely due to chance. So I'm wondering if you guys have seen, or done any research based on a set of observational studies, and it's all lead to similar results to a set of, clinical trial results. And like what you have said previously, even carrying out clinical trials, you can have different results from different clinical trials by looking at the same problem. Could you please expand about that? Thanks.
Thank you. Yes, please.
Yeah, I was just have a follow-up question to, whether observational study can lead to a similar result to a clinical trial.
think along the lines of the work I was describing by FDA, absolutely, you need a lot of experience to understand where clinical trials can be potentially replaced by FDA, by RWD. And so that volume of 30 trials for initial decision making, and perhaps looking into phase 4 trials as well, will give us that body of information. I think taking a step back, two things. First of all, I think we need to understand this for regulatory decision making, because there's a real limit to the number of clinical trials that we can run. Just the amount of effort, investigator time, pharmacist time, and so on that goes on to create an excellent clinical trial is a real limit. And in the United States, patients who participate in clinical trials are meaningfully different in many instances than the general population of patients who are treated with a drug. So it's imperative to understand where we can use other kinds of data, and I would be a proponent of real-world data and real-world evidence to really gain the broad understanding among all the patients treated with the drug, in important subgroups of patients treated with the drug, and to answer the range of questions that we have about a drug that very likely can't be feasibly addressed for one reason or another in randomized trials at all times.
And I would just like to echo some of those points, and one of the reasons for highlighting in this presentation about the utilisation going forward and capturing that real-world experience and real-world data is because the concomitant medications also changes across time. So when the clinical trial might have been done and certain drugs might have been excluded, 2 or 3 years on, and I think we're seeing this with our cardiovascular drugs and in diabetes, the combinations that people are taking is changing. And so I think this is a really important factor to be considering as well when we look at this. And in our case in Australia, where we're looking at the utilisation and the changing patterns of use, it gives us some indicators as well about the co-administration of other drug therapy at the same time. And I think this is one of the things in the clinical trials, and certainly is looking at the concomitant medication use, because that is changing rapidly now in certain areas in oncology, the biologics, and in the cardiovascular, as I said, and diabetes area in particular. And without that real-world data, we won't get that experience of the concomitant use of medications. And particularly, I think, as we move increasingly to the biologics, and what that will mean going forward in terms of understanding real-world use.
I think we have a question from over the left-hand side of the audience. Still? Oh, a question in the back there, please. If you keep your hand up so the mic can come to you. Thank you.
Hello, my name is Hanol John. I'm from South Korea. My question is for Dr. Rasen. Can you tell us some examples of studies that were used as RWE by using RWD on biopharmaceuticals in the US?
I think there's a large number of such examples just from the presentation this morning. The tocilizumab example was a study that was submitted to FDA and used for decision making by them. I think, I don't know if you recall, but there was a slide with RWD and RCTs and a relative a priori confidence. And I think the cases that were most common in the US are those cases for using RWE for post-market safety evaluations. So that would be the most common case that I've seen. Andrew, do you?
No, I think you're right. I think the common uses at the moment, at least from regulators, are in the safety world. But I think you did highlight, which is the other area which is growing a lot more now, which is the sort of concomitant arms in clinical trials. If you've got single-arm studies, rare diseases, oncology, and using historical or contemporaneous controls, that's probably the other big area. And I think FDA and other agencies around the world are thinking through this whole, what could you do in the label expansion, line indication expansion, sort of areas where pragmatic trials, observational database studies, others could come into play to be able to use it more frequently. And I think the rest of the challenging point is probably what a couple of people have raised is, when do you believe the observational data versus the clinical trials? And from an FDA language perspective, what is substantive evidence? It's always been interpreted as two double-blind randomized clinical trials, plus or minus a little bit. Where, if you start to think about real-world evidence and observational studies, how do you get that acceptability and that definition of substantive evidence?
The only other comment that I would make is also from a payer's perspective, is if when you look at who the drug is being used in, is it still cost effective in that population as well? So both from a safety, but also in terms of the value that is being returned for that.
Thank you. And just one final thought. There was an interesting article in the Wall Street Journal maybe a month or 2 months ago, which I think is probably not news to most of the people in this room, but it was about the just intense difficulty of recruiting patients for clinical trials. And I think your first slide with numbers along the lines of 3 and 7% spoke to that. And so I think we as a field have a real imperative to understand how we can really answer the questions we need answered with different methodologies. And I think recruiting patients to clinical trials is difficult for many reasons. Location and treatment was the example I believe you gave, but trial design and inclusion criteria, exclusion criteria, sort of qualification for patients to participate in the trial. These are major barriers to understanding how we need understanding the information we need to make clinical decisions and to make decisions about value.
Thank you.
Professor Park, and then the lady behind. Have we got the microphone, please, over?
Thank you very much. Very interesting and very important issues. I think that self-lung study is very, very difficult one. I think it's possible because of the collaboration between the healthcare professionals, clinicians, and pharmacists, and sponsors, big investment. And we can use the link between the EMI cameras. It's very, very extraordinary situation. So I don't think it's feasible for other countries and other places. So the strategy is that the real world we are using is very cost-effective approach to produce real world evidence. So I want to know about the current situation of regulatory agencies. The regulatory agencies' attitude is changing to accept real-world evidence to prove their secondary indication, especially in the case of EU and US FDA and Australia and UK. Because I want to say to our Korean FDA and other China, including Asian countries, to make attitude change for accept real-world evidence. as well as RCT result. So what is the current situation for advanced countries?
Thank you very much for that question. I mean, I can start off a bit on the side of the EU. So the two examples I gave are where the EU regulators for a primary indication accepted that you could not do an RCT, and they accepted a single arm with various sorts of controls, either historical or from a registry. And I also know of an example with a multiple sclerosis drug where for an extension of indication they accepted data solely from a registry or databases, I can't remember which it was, to extend the indication into the paediatric population from the initial authorization that was adult. Now, it wasn't strictly, you might say technically get a paediatric indication, but the wording was, it took away the statement that said there's no experience of use in the paediatric population to saying that there's experience of use in real-world evidence that suggests that the efficacy and the safety would be very much the same. So I think there's gradually much more increased acceptance from them, and also the realization that in some areas you just cannot do an RCT, or you really don't want to. What about the experience from?
I think the UK experience mirrors Europe on the whole. Most medicines in the UK are authorised through the central procedure in Europe.
At the moment.
At the moment. That might change in a few months' time as we leave Europe. But there are very few where you would have a uniquely singular market authorisation in the European territory at the moment. But I think we've certainly licensed one of our drugs that we actually divested a few months ago as an ultra-rare condition where we did a licensing on essentially 18 observations of patients who've been treated. Oncology, I think, for anyone who argues that a single-arm clinical trial is a clinical trial, it's not. It's an observation study of a group of people treated with an agent. So a lot of oncology studies are approved essentially in the real world, the usual care settings. It's really, I think the distinction there is the nomenclature we use to define how we collect the data. So we call it a clinical trial because we have a case report form and we bring the data in. We could equally call it a registry, and it would be and suddenly it would hit the real world context instead of the clinical trial context. So I think there's a lot of examples of use. I think what we've probably all been saying is there aren't that many examples of primary use, of primary regulatory approval for a novel indication or a novel medicine in a novel class of action. That I think is a long, long way away, and I think is always going to be a randomised trial, in my opinion.
But your exception was Strimvelis, wasn't it?
Yeah.
Where you had, it was on the basis of 17 patients treated, and because every other patient with the thing died, but at a very, very early age, and these young children survived, you have pretty good proof that it was
no trial that you
yeah.
could do. Yeah. And the FDA actually, for that same agent that Europe accepted it for, they asked us to do a clinical trial, and we had to go back to them and say it would take us 65 years to actually recruit sufficient patients into the clinical trial, because it's such an ultra-rare and ultra-orphan condition that you can't do a clinical trial.
So in the United States, I think FDA, like EMA, perhaps MHRA and others, have really, let me take a step back. It's hard to ignore the experience that's being recorded in real-world data as a source of information about decision-making, and it's ultimately the question, which decisions can we make, and in what cases can we make them? And I want to be very clear, in which cases can we not make them? That FDA is very actively trying to understand, and that's both, I think, their own interest in understanding that, and it's also part of legislation passed by the US Congress that says that they need to develop that point of view by, I believe it's 2021. That's the 21st Century Cures Act and the PDUFA 6 Act. But I want to sort of bring it to the topic of ISPE. Just one final thought, which is I think we're in this position where we can now use or think about using real-world data for decision making because of the work of ISPE and the chapters of ISPE and the work of all the people in this room on how over the last 20 years we've really developed methodologies that are very robust, that we understand well where they work and don't work, for working with the limitations and the biases available in real-world data so that we can now be thinking very actively about how to use this information we've collected for decision making. And I think this is really very much reflective of the work here in this room, the work that we saw in Prague,